What if you could rewrite the very code of life? Not just for one organism in a lab, but for an entire species out in the wild. We're talking about a technology called CRISPR-based gene drive. And, it has the potential to solve some of the biggest problems humanity is facing, from horrible diseases to out-of-control invasive species. So yeah, let's dive into this.
Just to get a sense of the scale here, let this number sink in. 263 million. That's the estimated number of malaria cases just in 2023. It's a massive, devastating global health crisis. And it's exactly this kind of problem that has scientists searching for a game-changing tool. And that tool? Well, it might just be gene drive.
So what is this incredibly powerful technology? Well, in this first part, we're going to break down how CRISPR gene drive basically acts like a cheat code for genetics, a way to make sure a specific gene spreads through a population faster and more effectively than nature ever intended. Okay, so at the very heart of all this is something you've probably heard of, CRISPR-Cas9. The simplest way to think about it is like a find and replace function for DNA. It uses a little piece of RNA as a guide to find a super specific spot in an organism's genes, and then this enzyme, Cas9, acts like a pair of molecular scissors and just snip, makes a perfect cut. This lets scientists edit genes with unbelievable precision. Now, this technology didn't just pop up out of nowhere. The whole journey really started way back in 1987, when scientists found these weird, repeating DNA sequences in bacteria. But the big aha moment came in 2012. That's when researchers figured out how to take this natural defense system from bacteria and turn it into a tool that we could program to edit any genome. It was so revolutionary that by 2020, the scientists behind it were awarded the Nobel Prize.
All right, this is where it gets really mind-bending. CRISPR itself is the tool, the scissors. But the gene drive part, that's the engine that uses CRISPR to completely break the normal rules of inheritance. Let's take a look at how that engine actually works.
Normally, in high school biology, we all learn that any given gene has about a 50-50 shot of being passed down to the kids. It's a coin toss, right? That's classic Mendelian genetics. But a gene drive completely hijacks that process. I mean, look at this slide. Instead of a coin toss, a gene drive gives a trait an almost 100% chance of being passed on. In some of the experiments they've done with mosquitoes, they've seen rates as high as 99.6%. That's just wild. So how on earth does it do that? It's a process called homing. So imagine you have a pair of chromosomes. One has the gene drive, and the other is just the normal, wild version. The Cas9 enzyme, which is part of the drive, finds its matching spot on the normal chromosome and, snip, it cuts it. Now the cell freaks out a little because it has a broken chromosome and it needs to fix it. So it looks at the other, intact chromosome, the one with the gene drive, and uses it as the template for the repair. And in doing so, it copies the gene drive over to the other chromosome. It literally turns an individual with one copy into an individual with two copies.
I know, it sounds almost too perfect, right? A system that can spread any gene you want through an entire population in just a few generations. But, as you might expect, there's a pretty big catch. Evolution has a funny way of pushing back.
This is the question that's been on every scientist's mind from the very beginning. If this is so powerful, what could possibly stop it? And the answer, ironically, is hidden in the very same process that makes it work in the first place, that whole cut and repair thing. This quote from the research really says it all. The single biggest threat, the Achilles heel of this entire technology, is the evolution of resistance. Okay, so here's the deal. Remember how the cell repairs the cut DNA? Well, that homing process, where it copies the drive, is the ideal way. But sometimes the cell just takes a shortcut. It uses a sloppier, quicker repair method that just mashes the broken ends back together. And this sloppy repair can create a tiny little typo, a mutation, right at the spot where the drive is supposed to cut. And if that happens, the guide RNA can't find its target anymore. That chromosome is now permanently immune to the gene drive. And just like that, you've got resistance. Now, this whole resistance problem could have been a total dead end for the technology. But instead, it just sparked this amazing wave of creativity, with scientists designing even smarter ways to basically outmaneuver evolution itself. So to fight back, they came up with this really cool multi-part strategy. First, instead of using just one guide RNA to make one cut, they use several. It's called multiplexing. Second, they target genes that are absolutely critical for the organism to survive or reproduce. And third, and this is super clever, they build a backup copy of the gene they're targeting right into the gene drive itself. This diagram really shows you how brilliant this is. By using multiple guide RNAs, you make it way, way harder for resistance to pop up. Think of it like this. A single mutation might block one of the cut sites, but the drive can still cut at the other locations. It's like needing 3 different passwords to get into an account instead of just one. As long as one of the cuts works, the drive can still get copied over. And what's more, making multiple cuts can just delete the whole section in between, making sure that if any resistance does appear, it's in a totally broken, non-functional gene anyway.
And hey, this isn't just a cool idea on paper. It actually works. Experiments have shown that just adding a second guide RNA can dramatically boost the success rate of the drive, and at the same time, lower the chances of resistance forming. It's a proven way to make these gene drives tougher and more effective.
So, we've seen how gene drive works, we've seen how scientists are making it even better, but all of this leads us to the biggest questions of all. And they're not really about the science anymore. They're about the huge ethical and ecological responsibilities that come with having this kind of power. This image, I think, captures the core problem perfectly. A gene drive is designed to be invasive. It's built to spread. So once you let it out of the lab, how do you ever get it back? How do you keep it from spreading beyond the species or the island you were targeting? These are the questions that keep ecologists and ethicists up at night. I mean, on the one hand, the potential is just incredible. We could maybe get rid of invasive rats that are wiping out native birds, or we could stop mosquitoes from spreading malaria. But on the other hand, what we're talking about is creating wild GMOs, genetically modified organisms designed to permanently alter nature. There's a very real risk of unintended consequences, of causing what some have called an ecological cacophony with unpredictable long-term effects. And that's why there's so much research right now trying to create drives that can shut themselves off or maybe even be reversed. And really, that all brings us to this final massive question. For the very first time in history, humanity has a tool that could be used to intentionally drive a species to extinction. The question is no longer, can we do it, but should we? And there is no easy answer to that. It's a conversation we all need to be a part of as this incredible technology gets closer and closer to becoming a reality.